Phoebe Lewis, a supplemental instructor for Dr. Byron Quinn's Natural Science Biology 1 course at Langston University, and I've compiled a series of review videos to help you with your final. This review video focuses on Chapter 11, which gives us an introduction to genetics. So what we'll be covering in this video, we'll be covering some history over Gregor Mendel, we'll be covering dominant versus recessive traits, we'll talk about phenotypes that Mendel studied, we'll talk about key terms in genetics, how to make a monohybrid cross and a dihybrid cross, and then incomplete dominance and codominance. So first, let's discuss Gregor Mendel. Gregor Mendel is considered the father of Mendelian genetics, and he studied the pea plant. Gregor Mendel was a monk, and like I said, he studied the pea plant. He looked at several phenotypes in the pea plant and observed how different traits were being inherited. So first, let's discuss and clarify dominant versus recessive traits. In the case of the pea plant, the yellow seeds were dominant to green seeds. So all that means is that the dominant traits always show up. Recessive traits only showed up when paired with another recessive gene. So what Gregor Mendel did was he crossed a plant with yellow seeds with a plant with green seeds. Now remember, the yellow seeds are dominant to the green seeds. What he found was that all of his offspring produced yellow seeds. So the green seed, the trait for green seeds, just totally appeared to just totally seemed to disappear. And so the cross is over here on the left hand side, the Punnett square cross. As you can see, if he took plants that were yellow, big Y, and crossed them with plants that were green, little y, all of the offspring had the genotype big Y, little y, which showed up to be all yellow seeds. So all offspring showed up yellow because the yellow is the dominant phenotype over green. So you can see the yellow is the dominant phenotype, where the green is the recessive phenotype. So if we look here, these were the actual pea plants he studied. What two phenotypes are observed on this slide? Well, he looked at seed color, so you can see this yellow in the green seeds. He looked at seed color, but he also looked at seed texture. So you can see a green wrinkled seed right here and a smooth seed right here. So wrinkled and smooth and yellow and green were the two phenotypes that are observed on this slide. So all in all, Mendel studied several phenotypes on the pea plant. He studied the seed shape, the color, the pod shape, the pod color, flower position, and the stem length. So these were all phenotypes that Mendel studied during his lifetime. So first, let's cover some key terms in genetics. First, the term phenotype. That refers to the physical characteristics that you can observe. Genotype refers to the genes responsible for the phenotype. Dominance refers to a phenotype that always shows up. Recessive, a phenotype that only shows up when paired with another recessive gene, and it will be masked by a dominant gene if one is present. Homozygous says that both genes are the same, so either both are dominant or both are recessive. Heterozygous says that each gene is different, one dominant and one recessive. Alleles are just simply alternate forms of genes, so you can either have a dominant form or a recessive form. A monohybrid cross is a Punnett square that's looking at one phenotype. Dihybrid cross is a Punnett square that looks at two phenotypes. The phenotypic ratio is the ratio of observed phenotypes, where the genotypic ratio is the ratio of the genotypes in the offspring. So you might want to take a moment to pause this video, write this down, you might want to go ahead and write that down. Each of these, make yourself some flashcards so that you're able to remember what each of these terms mean, because we're going to be using them a lot in this chapter. So just one note, remember, alleles are alternate forms of genes, and they're located on the chromosome. And so you get each of your chromosomes, remember, you get one from your mother and one from your father. So your mother gives you one allele for a particular trait, and your father gives you another allele. And so if both your mother and your father give you the same dominant allele, you'll be homozygous dominant for a particular trait. If your mother gives you a dominant allele and your father gives you a recessive allele, well, you'll have the dominant phenotype, but you'll be heterozygous or a carrier for that trait. If your mother and your father both give you the recessive allele, you'll display the recessive phenotype, and you'll be homozygous recessive for that trait. So let's talk about how to demonstrate a monohybrid cross using a Punnett square. So let's take, for instance, the mother's genotype is heterozygous. She has a big B and a little b for whatever particular trait. We can say brown hair in this case. Big B, little b. So she has brown hair. She's heterozygous for the trait. The father is also heterozygous for the trait. Let's say big B, little b, he has brown hair as well. Well, in order to make your monohybrid cross, you first have to figure out what particular alleles are going to go on the gametes. You do that by basically breaking the genotype in half. So a big B will go right here, a little b will go right here. You do the same with the father. The big B goes right there, the little b goes right here. And then you're able to just take those and fill in the square as follows. So the big B makes a big B, big B. The big B, little b will make big B, little b. Big B, little b again makes another big B, little b. Little b will make a little b offspring. And so what you're able to do is then find your genotypic ratio. You have one homozygous dominant, which is displayed right here. Two heterozygous, which are displayed right there. One homozygous recessive, which is displayed right there. And remember, we're asking the genotypic ratio, so that gives us a grand total of 1 to 1. 1 to 2 to 1. And you should see that that adds up to 4. 1 plus 2 is 3. 3 plus 1 is 4. You have 4 offspring right here. We've accounted for all genotypes. One homozygous dominant, two heterozygous, 1 homozygous recessive. Well, the phenotypic ratio is a little bit different. We're just looking at the phenotypes that we see. You can see here that you have three of these offspring display the dominant phenotype, those 3 right there. And one of them displays the recessive phenotype, that one right there. And so that gives us a phenotypic ratio of 3 to 1. Again, that adds up to 4. And so one thing that you need to notice is that 3 genotypes, you have 3 different genotypes. This is one genotype, these 2 is another genotype, and this is the third genotype. And they produce only 2 different phenotypes. So what the key thing to realize is that big B, and big B, little b produce the same phenotype. So you're not really able to know if you just know the phenotype and don't know the genotype. You don't know if they display the dominant phenotype, whether they're heterozygous or homozygous dominant for that genotype. And so you have the 3 different genotypes. They produce 2 phenotypes. So that's one thing to know. Another thing that you can do is a dihybrid cross. Now, a dihybrid cross, if you look at your key terms, is basically a Punnett square where you're looking at 2 different traits. So in the case of Mendel, he looked at not only the seed color, whether it was yellow or green, but he also looked at the seed texture, whether it was smooth or wrinkled, as we discussed earlier. So this was his dihybrid cross. And I know a lot of you all get confused on that, so I'm going to take you step by step on how to make a dihybrid cross. So how to demonstrate a dihybrid cross using a Punnett square. So let's just use the Mendel's example. And we had the maternal genotype being big S, little s, big Y, little y. And the paternal genotype being big S, little s, big Y, little y. Same. Well, you guys have done algebra before, and you know how to FOIL. First, outside, inside, last. So you're foiling to find your gametes. Now remember that on top of each box, since we're looking at 2 traits, you need to have 2 alleles on top of each box. So first you take the first. So you have a big S and a big Y. And that you will put that on top of your first box like so. Then your outside, you have big S, little y. Put that on top of your box like so. Inside, little s, big Y. Put that on your box. Little s, little y. Put that on your box, okay? And then the same thing with the paternal genotype. You just go through first, outside, inside, last. And then just like you did in your monohybrid cross, you basically take what you get from each side and pair them together to see what your offspring's genotype would be. So your offspring's genotype should have 4 alleles, just like your parent genotypes. So you would have big S, big Y, big Y. In this case, you would have big S, big Y, little y. In this case, you would have big S, little s, big Y, big Y. In this case, you would have big S, little s, big Y, little y. Big S, big Y, little y. Big S, little y, little y. Big S, little s, big Y, little y. big S, little y, little y. Big S, little s, big Y, big Y. Big S, little s, big Y, little y. Little s, big Y, big Y. Little s, big Y, little y. Big S, little s, big Y, little y. Big S, little y, little y. Little s, big Y, little y. And little s, little big Y, little s, little y, excuse me. And so that's how you get your dihybrid cross. You see, notice that there are 16 boxes here. And then just for format, you always put the like letters together. So you always put your S's together, put your Y's together. If there is a dominant gene, you always list that one first. If there, and so that's just a little formatting note. So our phenotypic ratio, what we noticed is that we have 9 that are smooth and yellow. So these 9 are smooth and yellow. Then we have 3 of them that are wrinkled and yellow. The wrinkled and yellow ones are right there. Then you have 3 of them that are smooth and green, that are shown right there, and 1 of them that is wrinkled and green. And so that's where you get your infamous 9331 phenotypic ratio that you learned in lab before. So 9331, this is how you get it. Just some little side notes. We'll briefly discuss incomplete dominance. This is actually exceptions to Mendel's rules. So incomplete dominance occurs where you have a red flower and a white flower, and they actually blend together and produce a pink flower. So not all the times, in some special cases, you'll have a blending of traits, which is an exception to Mendel's rules. So a red flower and a white flower making a pink flower. You get that blending. That is an example of incomplete dominance. Codominance occurs where both, like an example of this would be on your blood. So if you're type A blood, type B blood, type AB blood, or type O blood, that is a perfect example of codominance. That occurs when both traits show up together. So in the case of AB would be the perfect thing. Your mother may be type A blood, your father is type B, and you come together and you're type AB. So you have a codominance, or both of them show up. So that covers chapter 11, and stay tuned for chapter 12 through 15.